Good morning, everyone. Once again, my name is Diego de la Torre, and I'm from Oral Roberts University. Today, I'll be presenting on cancer cell cytotoxic effects of compounds secreted by Canadenum luteolum. So for this presentation, I'll be going over my thesis, background, evidence, and followed by discussion. So the thesis. So the goal of this project was to find a naturally occurring product that not only kills cancer cells, but keeps your normal cells alive. So I was able to find a product like this. And then the next part of the experiment was to isolate it and to find what concentration is it best useful at. So for my background, I'll be going over cancer, mushrooms, cell culture, followed by the NGC chromatography system. So what is cancer? So many of you guys are accustomed to cancer. You have a lot of family members who might have had cancer at one point. So a basic definition of cancer is it's uncontrolled cell division or growth. So this is where normal cells are changed in the regard that they are made to grow uncontrollably and there's no way for your body to naturally suppress it. So cancer has these 10 hallmarks or 10 traits that allow them to be called cancer. So one of them is that it evades growth suppressors such as tumor repressors. It also evades cell death and evades the immune system. So the problem with cancer is that it's your own body. So your immune system isn't really equipped to kill your own body because it's there to protect your body. So that's why cancer is so dangerous. Another problem with cancer is that there's no definite cure. So there are various treatment options. So one of the most popular treatment options is chemotherapy. So chemotherapy not kills the cancer cells through radiation, but the problem with chemo is it kills cancer, but it also kills your normal cells at the same time. The only reason we keep using it is it kills yourself a bit slower. So a lot of people actually die due to chemo versus the cancer it's trying to kill. So the main cancer I'm focused on is breast cancer. So breast cancer is one of the most diagnosed worldwide. So back in 2022, 670,000 people died due to breast cancer. And the specific breast cancer I'm focusing on is MDA-MB468 cancer. So this is a triple negative breast cancer. And the reason it's called triple negative is that it has 3 receptors that are missing. So these receptors allow your immune system to kill them more effectively, or there are different treatments that allow cancer to be killed when it targets these receptors. But this cancer is missing these receptors. That's why it makes it one of the most malignant or dangerous ones to have. So cell culture. So it's not very ethical to make a compound or make a drug and then give it to a human right away. So we had to test the drug in the laboratory setting. So this is where cell culture comes into hand. Cell culture is a way to have cells in a laboratory. This is where a human who had cancer actually donated their cancer cells to a research facility, and then they were able to transform it to survive in a laboratory setting. So cell culture is where you have a plastic dish and you grow the cells in there in one layer with growth media. And this media provides the environment to grow. So it has all the nutrients and growth factors, plus it has a lot of conditions such as like normal pH and temperature-wise to grow cells. So another background information is mushrooms. So most of you guys are accustomed to eating mushrooms. So mushrooms have been used for thousands of years for medicinal or just a food source. So one of the oldest human remains was found to have mushrooms in their pockets. So this just shows mushrooms have been used for thousands of years. And due to this, our body is accustomed to eating it. So there's not major allergic reactions to eating mushrooms because we've been accustomed to eating them. So, and then followed by this is the NGC chromatography system. So this fancy machine allows you to identify what's inside the solution and to separate it by size. So what this basically does is I add 1 ml in and it dilutes it into 26 milliliters. And through this process, it helps me know what's in there and it separates it for me to test on different cancer types. So why this project? So I chose this project several years ago because Dr. Ranahan has been using mushrooms for research purposes for years. So I just tagged along because they were having great results. So I decided to join the project and to give more data on why mushrooms are a good alternative for medicinal use. So my project involved growing the cells, isolating the drug, identifying using the NGC system, testing the drug against cancer cells, and then analyzing the results. So mushroom growth. So most of you guys are accustomed to eating the fruiting body of a mushroom. So this is what is above ground, but below ground is something called mycelia. So this is where most of the mushroom is actually at, below ground. And what we do for our lab is we grow the mycelia on agar. So agar is basically gelatin. So when it's hot, it's liquid form. When it's solid, when it's room temperature, it becomes solid. So this provides a great environment to grow mycelia. It allows us to grow what's supposed to be underground, above ground to use for our experiments. So what I did is I made agar and then I added the mushroom on there. And this mushroom is white. As you can see, it's a bit fuzzy and overgrowing the mushroom plate. This just shows that this agar is a good environment for the mushrooms to grow. And then after the mushroom was fully grown, I added water. Water was used as a vehicle to gather the drug. So the drug we're using is called secretions for mushrooms. So mushrooms don't have a stomach. So what they do is they give off enzymes or chemicals that break down what's around them in their environment so they can then grab the nutrients. So what we're doing is we're getting the actual secretions. Most current treatments that use mushrooms as medicine use, they're using what's inside the mushroom. What we're using is what the mushroom makes to break down the environment. So this is a new form of research Dr. Rain had developed. And then after I grabbed the water that had the secretions inside of it, I then concentrated to make a 10x solution to make it more easy to handle. So followed by this is I then isolated using the NGC chromatography system. So as I mentioned earlier, this machine separates a compound based on size. So what I did is I added 1 ml and then this machine diluted it into 26 mils. So here's only part of the data. So on top left, it's 8.6 and on top right is 8.24. So from 8.6, that's where the biggest molecules come out. 8.24 is the smallest molecules. And this just gives us a range for me to grab specific areas I want to test. And there are some fancy colors here. The blue is peptides. The green is proteins, while the orange is polysaccharides. So this machine uses wavelengths to identify what's inside the solution. So the point of interest I looked at was 814 right here. So 814 has a peptide, which is shown by the blue, has a protein, which is shown by the green, and a polysaccharide, which is shown by the orange. So I tested all of this in my experiment, but I was mostly curious on what 814 did. So for the actual testing of the drug, on the first day, I added the cancer cells into a 96-well assay. So this is just a plastic dish that has 96 wells compartments. So these compartments allow me to have the same condition, but test different drugs at the same time. On the next day, I replaced the media with Optimum. So normal media has all these nutrients and growth factors to have the cells grow super fast. But our body is not completely filled with growth factors. To replicate the body a bit more, I replaced the media with Optimum. So Optimum has all the nutrients, but it's missing all the growth factors. The next day, I added drug or vehicle. So the drug was the previous image I shown. I just added it to different compartments, and the vehicle was water. So the reason why we have a vehicle is as a control to show that the drug actually killed the cancer, not my inability of actually working with the cells. The following day, I added Abcam, which is a compound that helps me visualize the metabolism of the cells. So it doesn't really tell me how many cells died, but it does tell me how active they are, which we can use as a point of reference. The following day, I added the plate into a plate reader, which then gave me information to analyze on what came out and how active the cancer cells are. So here's an image of where I place the cells and where I place the drug. So each 10 quadrants right here is one specific condition, so one specific drug. So the top row is a vehicle, so this is just water. Bottom row is the drug at that specific concentration. So this is done 5 times, 5 replicates to show it's not due to error. And right here, this is one quadrant. There's another quadrant right here, third, fourth, fifth, and sixth. So in this one experiment, I was able to test six different drugs. And the blue right here is just more wells that show me background reading. So here is the numbers that the machine gave me. The machine looks at AbChem. So AbChem is a reagent that is consumed by the mitochondria. So the mitochondria is what gives the cell energy. And if a cell doesn't have energy, it basically dies. So this AbChem is right at 605 wavelength. And when the mitochondria consumes it and makes a byproduct, it's right at 570. So this machine gives out numbers based on what wavelengths it absorbs and what's basically happening in there. So this is very complicated. So I made an easy bar graph to understand. So on the far left, there's numbers from 0 to 120. So this is percent viability, where 100 is just means it's a control where the cells did not grow or did not die. It's just a way where the cells already had water, so there was no experimental value in there. So what we want is the data to show it's below 100. And as you can see, most of this data is below 100. On the second bar graph right here, it's actually around 70. So this is 70% viability. What this means is the cancer cells are 30% less active compared to regular cells. So they're less active. That means it could be that they're more dead or that they're just having less energy to proliferate. So once again, 814 is where I was looking at. And this is right here, this bar graph. So this shows that our compound is a peptide-containing polysaccharide since it has peptides, proteins, and sugars, which is polysaccharides. So the main thing about this experiment was finding a drug that can kill cancer. Well, what's the use of having a drug that kills cancer but kills you? Might as well put bleach or gasoline on yourself because that kills the cancer, right? So what I had to do next is test the same drug on both cancer and non-cancer. So this is what this experiment is on. The dark blue represents cancer cells, while the light blue represents normal mammalian cells. So on the far left, we have the same values when it comes to viability, 0 to 180. So these are put in groups of 2. So there's the first set, second set, and third set. So these groups had the same drug and the same condition. So on the first 2 bar graphs, the first 2 bars, the left one is the cancer cells. This is around 75% viability, while the one on the right is the non-cancer, 140% viability. So what this data suggests is not only did the drug decrease the viability of cancer cells, but actually gave a huge boost to non-cancer cells. So this drug kills the cancer, but it provides nutrients for a normal cell to grow. Compare that to chemo, where it both kills the cancer and it kills normal cells. This is a great alternative. So this is just on one condition. And these are several other conditions where I was testing out different data and different concentrations to see what is the most optimal state. And on the middle here, this kills the cancer even more, but it doesn't provide as much nutrients to the regular breast cells. So what happened with this experiment? So this experiment, I was able to isolate a compound that killed cancer, but not kill the normal cells at the same time. So this is amazing since we've had a lot of treatments that just kill cancer and kill humans at the same time. So the amazing part about this is that this mushroom is naturally growing. So we found this mushroom online, which was non-GMO. So this is just a mushroom that's found in the wild. And the great thing about this experiment is that this can be done at home. The mushrooms can be grown at home. All you need is a pressure cooker to sterilize the nutrients and the environment for the mushrooms to grow. The problem with this experiment is that there's natural variability. So this natural variability is due to the mushroom being an alive organism. So since it's an organism, we can't control what products it creates and how active it is. And there's just a lot of variables we can't control. So that would be a way to future research is to try to reduce the variability. So another problem with the experiment was that this was grown in a laboratory setting. And as you guys know, laboratory and a human body are completely different. So a way to have a good intermediate is matrigel. So matrigel allows the cells to grow tissues and to go into 3D environments. So this is where we can grow both cancer and non-cancers in the same place. So this allows us to test to show that in the same environment where the cancer and the normal cells are next to each other, this will kill the cancer and make your normal cells grow more active. So I've actually been testing on this experiment, and we recently had to move labs. So this was completely on hold. So this summer I'll be finishing this up and hopefully I have great results. So here's some work cited, and are there any questions?
Explain that last slide with matrigel. What's different about that? Why are you doing this?
Okay, so in the normal experiments, when I tested the drug against cancer, it was in a one-layer environment. So it was just one layer of cancer cells and one layer of normal breast cells. But you guys have bodies, you can tell there's thousands and millions of layers. So this isn't like really how the body is when it's just one layer. So matrigel allows us to form these cells into 3 environments, so it creates multiple layers. And that way we can test the drug in a more environment that's more suitable to how humans are.
I know that you use water as a means of diluting or whatever you, however you explained it. Was the water deionized, radioactive? Was there anything special about the water?
So this water is deionized and then sterilized using a millipore machine.
Would there be any benefit to using some heavy water with a carbon, an isotope that's radioactive?
We haven't tested that because if we added more variables, it just creates more data points that can go wrong. But we can definitely do that going on where we test different waters to extract the solution and see if it's even better.
Thank you very much.